So a very good evening to our esteemed guest, our valued audience, and my dear colleagues. My name is Yasha Nirati, and I currently hold a position of a scientist at Biocon Limited in Bangalore, India. On behalf of Neighborman Bioinformatics, I'm thrilled to welcome all of you to today's webinar, where we will delve into the captivating world of 16S metagenomics as we explore how the QIIME2 pipeline demystifies the intricacies of microbial communities. This session is designed to provide insights into fundamentals of metagenomics, offering a practical walkthrough of 16S rRNA gene analysis using QIIME2 pipeline. So please allow me to provide a brief introduction to our Neighborman Bioinformatics. Founded in 2019, Neighborman Bioinformatics is a globally renowned bioinformatics service firm that boasts a unique and far-reaching vision. Our mission is to establish an encompassing network that brings together academia, industries, students, and active researchers from around the world within the expansive fields of bioinformatics, biology, and related subjects. The vision came into life with the launch of Neighborman Bioinformatics in Europe in 2021, extending exceptional training and services to individuals on a global scale. Our expertise is multifaceted and spans a wide range of disciplines, including omics, structural bioinformatics, computational drug design, molecular modeling, molecular dynamic simulation, cheminformatics, programming, artificial intelligence, machine learning, systems biology, human health informatics, and intellectual property rights. Over the years, we have organized numerous impactful workshops and skill development programs in various bioinformatics domains, complemented by an extensive portfolio of 200+ webinars, each drawing an average of 300+ participants from India and beyond. We are dedicated to nurturing collaborations and providing opportunities for project outsourcing, consultation, freelancing, and customized training programs. For more information and to get in touch, we invite you to visit our website, www.neighborman.com. So before moving ahead, I'd also like to introduce the ReproducibiliTea Hands-on Lecture Series that convenes bi-monthly with a special emphasis on guiding participants through practical sessions aimed at reproducing established bioinformatics pipelines. This inclusive platform extends a warm invitation to individuals at all academic levels, whether pursuing masters or bachelor's, Ph.D. degrees, or conducting a research in the field of bioinformatics and its related disciplines. If you are enthusiastic about staying informed or would like to be a part of our series, we encourage you to subscribe and learn more by visiting our website at www.neighborman.com/webinars. To introduce our guest speaker. So please allow me to present our guest speaker, who also happens to be my junior from Institute of Bioinformatics and Applied Biotechnology, IBAB in short, from Bangalore. Please join me in extending a warm welcome to Maya Jayachandran, currently serving as an Associate Data Scientist at Happiest Minds Technologies Private Limited. Maya is a holder of a master's degree and possesses a profound passion for realms of bioinformatics and data science. Her current work involves active engagement in the application of multi-omics strategies, creating pipelines for their implementation, and incorporating AI and machine learning-based models as supplementary layers to her projects. Her dedication to embracing interdisciplinary approaches serves as a source of inspiration and harmonizes seamlessly with the ever-evolving landscape of data science. We consider ourselves extremely fortunate to have her expertise with us today. Let's extend a hearty welcome to Maya. Maya, very warm welcome to our webinar.
Hello, everyone. Thank you, Yasha, for that wonderful introduction. And a very warm good evening and welcome all to this session. I'm extremely glad that you'll have taken time out of your weekends and you'll have joined this webinar. So before I begin, just a few points, right? So what are we expecting from this particular webinar? Right. So I'll be covering, as just mentioned before, some of the very core concepts in metagenomics. Right. I'll be diving deep into, about the 16S rRNA sequencing based approaches, about the metagenomic shotgun sequencing based approaches. And also towards the end of the webinar, I'll be having a demonstration of the QIIME2 pipeline. Now, today, because of certain time constraints, I'll be covering the very core, commands in the QIIME2 pipeline, right? And hopefully, I hope that you all of you all get to learn something new and fascinating today from this session. So I'll just share my screen. Right. I hope my screen is visible and I'm audible to everybody.
Yeah, sure.
Right. So, yes, today, of course, we'll be exploring the world of microbes, right? We'll be diving deep. So, just an overview on just like what I mentioned, right? We'll be seeing what do you mean by the microbiome, right? What is its significance? Then we'll be going on to certain very basic concepts in metagenomics. Then we'll be seeing what are the certain differences between 16S rRNA amplicon sequencing and also the shotgun metagenomic approaches. Further, we'll be diving deep into the 16S rRNA gene structure in itself and how it has evolved into being a marker gene, right, for bacterial or any other organismic identification, right? Further, we'll be getting on to the concepts of what is an OTU and ASV. And further, after we cover all these basic concepts, towards the end, we'll be beginning with the QIIME2 pipeline. There'll be a live demonstration, and we'll go through what are all the steps in the QIIME2 pipeline, what are the different types of input file formats, and all the details within. And then in the end, we'll also have a small discussion regarding what are the applications of whatever we have covered in this particular session. Right. So before I begin, I would love to know how many of you all have heard or you're familiar with the term microbiome and you all are familiar with the concepts in this domain. Thumbs up reaction would do on Zoom. So I see a pretty few of you all have reacted a thumbs up onto this. Right. So to all those of you all who have never heard this term before, right? What do I mean by the term microbiome? So we all have heard of microbes, definitely, right? And they're omnipresent. They're present everywhere. Take it air, be it water, be it soil, right? They're present all the places around us, right? And also the human body is also teeming with microbes, right? Every inch of our body is actually covered with microbes, which is very fascinating to know, right? It's a very fun fact that, the total number of human cells in our body is, less than compared to the number of microbes that we have on our human body. That's how abundantly present they are, right? So, if they are so widely spread on us, definitely there has to be some important function that they might be contributing to our survival, right? There has to be some significance, right? And that is definitely what all the scientists in this domain have set out to explore, right? And all of this is just an effort to know the human body a bit more better, right? So that is definitely, when we speak about microbial genomics, that is definitely the focus here, right? What sort of information can we extract from these microbes and their genetic material, right? So I'll just show you a few pictures on how abundantly these microbes are present on the human body, right? So here you can see the very first GIF. You can see that that's a gut, right? That has been represented there. And you can see how all the microbes are, it's teeming with microbes there, isn't it? And towards the right, this is a very beautiful picture that has been taken from one of the Nature articles, and very beautifully represents how there are so many different kinds of microbes that even is present on our skin, right? So present everywhere, right? And also now the next thing that you can see, this is also one of my favorite images. So this is a microscopic image of the human tongue. Right, this is also one of the papers which were published under Nature. So they have stained the human tongue, and here you can very clearly see how each color is actually representing a different species of the microbe, right? So you can see blue and red and pink and yellow, and all of these colors are actually representing different species of microbes that are present on the human tongue, right? Such important contributors to even our oral hygiene, isn't it? So, I mean, I can't stop talking about the significance that these small, tiny superheroes have with respect to the humans, right? Be it in regulating our immune responses from the smallest of processes, take it, be it digestion or even our hormonal control, or be our moods, right? How we regulate our mood. All of this, they're all impacted by these microscopic, wonderful organisms, right? And very much a reason why we need to study them in depth, right? So now, how do we, when I speak about studying them, right? How are we, where are we going to begin, right? Now, to people who already have an idea regarding this, obviously we all know that we'll be growing these microbes artificially in the laboratories, right? We'll give them specific nutrients, specific media wherein they can grow, right? We culture these microbes, right? And we let them grow, and then we analyze their growth and how they impact, isn't it? So that is one of the basic techniques how we do. So I'll be showing that in the next slide. As shown here, you can see how one of the scientists is taking a plate and waiting for the microbes to grow. So this is one of the most traditional methods wherein we follow lab-based techniques, right? But if you go to see, this is not feasible all the time, right? Not all different kinds of microbes that we have on this earth, we can't grow them artificially in the laboratory. There are very specific requirements, very specific growth conditions that they might require, which we might not be capable of, bringing it up in the laboratory, right? We cannot facilitate that all the time. And that is where bioinformatics comes into the picture, right? These are computational approaches. So, you have a laptop, you sit on a desk, and now you're capable of analyzing these particular microbes and their genomes just sitting on one place. So that's how fast technology has grown, right? So you can see I have shown a comparison here clearly. Towards the left, we have traditional methods, and towards the right, you can see how we can isolate these organisms, collect their DNA, sequence them, and then we can analyze them using computational based approaches. Right. So this is broadly, if I, just understanding this explains metagenomics in itself. Right. So what do I mean? What do you mean by metagenomics? Right. So genome is nothing but referring to the genetic material, and metagenomics is nothing wherein you are, utilizing different modern genomic studies wherein you're going to analyze these communities of microbes. And that's how microbes tend to live, right? They always tend to live in communities, right? In their natural environments that they are written here, right? And we do not need to go through the traditional methods of isolating them and cultivating them in the laboratory, right? Of the individual species. So this is broadly the term of metagenomics, right? All that has been facilitated by the technology that we have today. Now, when we speak about metagenomic-based analysis, right, this is one of the omics technologies, as they say it. What are we looking at, right? So there are two important questions that we tend to answer using metagenomic-based approaches. Number one, what we're interested to know is who are the microbes that are present within the human body, right? Who are they, right? And the second question that we try to answer is what are they contributing to us? What is their functional role? within our human body, right? So these are broadly the two of the most important questions that we tend to answer using these metagenomic based approaches. So you can see here in the right image, you can see here, this is one of the first thing that they mentioned is phylogenetic classification. I'll be getting that to that later. So here we tend to answer who is there, right? We can see here bacterium A, B, X, right? They are the names of the bacteria or the organisms within us. And towards the right, functionally, we are trying to understand what is it that they can do within the human body, right? So broadly, this is what is the landscape of metagenomics-based approaches. I hope we all are on the same page. Can I have a thumbs up reaction for that? Yes. Perfect. So I hope that's perfect. We'll move ahead, right? So now, how does the 16S rRNA come into the picture, right? So when I speak about identifying who is there or which are the organisms that are present in us, right? So we need to have a unique identification factor, right? That will tell you that if, on the basis of this particular molecule, I can tell you what are the different kinds of microbes that are present within the human body, right? And one such molecule that we use for such analysis is the 16S rRNA molecule. So what is the 16S rRNA? Nothing. rRNA stands for ribosomal RNA. And I've shown a very picture here. You can see that this is the bacterial ribosome, one of the organelles that is present. And the bacterial ribosome has two subunits, 50S and 30S. Now, this particular 16S ribosomal RNA is actually present on the 30S subunit, right? And 16S rRNA has various features, which is enabling it to be used as a unique marker factor, right? If you'll see. So this is a 16S rRNA molecule here, as you can see. And towards the right, you can see this is the gene which codes for this particular 16S rRNA molecule. Right? Now this structure, this particular 16S rRNA gene, if you'll see, it has a very unique structure, right? You'll see here that there are something called as variable regions and there are something called as constant regions or conserved regions, right? So the ones which have been shown in a creamish color and they have been denoted by V1, V2, V3, these are the variable regions. Now, the name itself is self-explanatory. These regions will be differing amongst different species of the bacteria, right? So in different bacteria, this particular region of the 16S rRNA gene will have a lot of differences sequence-wise, right? At the genetic level is what I'm speaking. And the blue regions that you can see here are referring to the constant regions or the conserved regions. That is, evolutionarily throughout the different kinds of bacteria, these sequences tend to be overall similar across all the different bacterial species that you can identify. Right. So this is one of the very unique, marker genes that we use in taxonomic annotation. That is basically just complex terms. This is just to identify which are the microbes that are present within us. Right. So how do you sequence this? You can see here the 16S rRNA gene. We generally sequence it. Right. And all the output files that we get from the sequencing run are what we analyze using the QIIME2 pipeline. Right, we'll come to that later in a bit. So here you can see how you will be having a specific primer sequence. You'll have a sample barcode region, and you'll specifically certain regions. Now, you may not always sequence the entire gene in the whole. You might just sequence specific regions, say only V3 or V4, or just V5 and V6. So different pairs of variable regions is what will be sequenced specifically, and you have the primer sequences that you'll be utilizing here will be targeting the conserved regions, right? Because since they are same, you can have a universal primer, right? It will be same throughout for all the different bacteria. So that is where we, that is how the 16S rRNA molecule is utilized. Once we finish, we get into the QIIME2 pipeline and we understand what are the microbial species that are present within whatever sample that we're dealing with and how abundantly they are present, right? So that is how the QIIME2 pipeline comes into the picture. Moving ahead. Now the other approach, right? So when I speak about metagenomic analysis, here when I speak about 16S rRNA, right? So 16S rRNA is one gene of the entire bacterial genome that we are focusing, right? And on the basis of that 16S rRNA gene, I am identifying which are the microbes that are present in a particular sample. Now, this is not always the approach that is taken. The other alternative approach that we can take under metagenomic is called as shotgun metagenomic approaches, right? So here we are not going to deal only with one portion of the bacterial genome. Rather, we'll be taking the entire genome in itself, right? And then we'll be analyzing it, right? So this is a much more in-depth analysis, I would say, and also the resolution of the taxonomical classification, right? We come to know is much greater in this particular approach, in the shotgun-based approach. As you can see here, you take the entire microbiome sample. Here you can see, suppose I take it from the gut, I collect all the samples, I extract the DNA, sequence them. Now here I'm collecting the entire genome, which is what is different from the 16S rRNA-based approach. This is a very major distinction, which some of the people tend to miss here and there, right? And we'll be having multiple samples. We'll have the samples, we'll be having a quality control check and getting on to the further details. I'm not diving much deep into this, but this is just to tell you all that these are the two main approaches under metagenomics that we generally use. Now, another concept that I would like to cover, right? So this is the concept of OTUs and ASVs. Now, these are the terms that we generally use in these bioinformatic tools. Like now, today I'm going to be explaining about QIIME2. And the similar tool that we use is referred to as DADA2. So these tools, right, they generally tend to aggregate the samples together, right? They tend to cluster them together, and they'll be, they'll be analyzing them as a single unit, right? So that is where OTUs and ASVs come into the picture. One of the output formats of the QIIME2 pipeline, right? So it is generally said that ASVs have a greater resolution than OTUs. I'll tell you why. So say you have, these are the different sample sequences that you are analyzing, right? You say you take a fecal sample, you have these particular sample DNA sequences that you have extracted from them, and now you PCR amplify them. What do I mean by that? Just make multiple copies of them, right? Once I make multiple copies, now what I do is I try to align these sequences against each other, right? Align them and try to find out how similar are these sequences to each other, right? So when I do this and say I put a cutoff, these sequences have to be at least 97% similar to each other, right? So if these sequences are similar to each other, now I group them together as one operational taxonomic unit or OTU. Right? So now this becomes one way of analyzing the microbial sequences. And now I can say these microbes, how abundant they are, right? So the unit in which I'll be telling them how abundant these microbes are will be in terms of a OTU, right? So you can see how all the red sequences have been clustered together into one particular OTU. You can see all the blue sequences have been clustered together into one blue OTU, right? So this is just a very simple representation of an OTU. On the other hand, what is an ASV, which is also referred to as an amplicon sequence variant, right? So say the same way I have my sample sequences and I amplify them using PCR. Now what I try to do is when I align them, rather than finding out how similar these sequences are, I would like to check out how different these sequences are, right? So I'm going in the opposite direction completely. So here, if I find that the sequences are at least even one nucleotide or two nucleotides different from each other, I'm going to assign them into separate ASVs. right? So each sequence will be assigned into a separate ASV. So here, just by the definition or just by the concept in itself, you can understand how ASVs tend to provide a much greater resolution in terms of OTUs, right? Because if there are any minor errors present in the sequences because of the PCR amplification run, or you may have certain random insertions or deletions in the sequences, all of them will be, they'll tend to be clustered together into one OTU, which is erroneous, right? On the other hand, if there are such differences amongst the sequences, I'll be classifying them as separate ASVs, which will be a much greater resolution that I can have, right? So as of now, as of the current version of QIIME2 pipeline, the general output format is in the form of ASVs. And earlier, it was in the form of OTUs. Now they have increased the resolution to ASVs. So this is one of the output formats, right? So that is the basic, some of the basic concepts that I have covered in, in under metagenomics. Now we'll be diving deep into the QIIME pipeline, right? So what is this QIIME pipeline useful for? Like I said, it will be answering the question of who are the microbes that are present in a particular sample. That is why we use QIIME2. Now, speaking about QIIME2, the makers say that it's pronounced as QIIME2 and not QIIME. So you all are free to use whatever the pronunciation that you are comfortable with. And QIIME2 has a lot of steps, right? It seems to be a very complex pipeline, right? There are a lot of steps. Actually, I would see it as there are a lot of utilities which are available in the QIIME2 pipeline, right? So you begin with a very raw input format, say the FASTQ files. I hope all of you are familiar with the FASTQ file format. So you begin with the FASTQ file format and you end with really beautiful, beautiful visualizations, and you'll have a lot of statistical outputs as well that is enabled in QIIME2. Another feature of QIIME is that it is supported across multiple platforms, right? So there is a one Linux command line version. The other part about it is that it's also enabled in a GUI support, because not all of us are familiar with Linux commands. So the another version that they have is through the Galaxy software, wherein everything has been automated. You just need to input the files and it'll run the entire pipeline for you. The other part is also you can use it in a Python API format, which is a web-based format, right? Web application-based format. So there are different formats which are enabled in QIIME2. This is to increase its flexibility of usage, right, across all the different research scholars who are utilizing this particular tool. And also another thing is that it has a very beautiful documentation. You are all free to check it out. So I'll be going on to that web page in a minute. So a very beautifully documented web page. You all can a very detailed explanation of all the different commands are also available, right? So let's before I get into the steps, I'll just show you all the web page, right? So you have to just enter QIIME2, and this is the page that pops up, right? So this is the documentation for QIIME2 for the latest version. Now you can see here getting started, and now one of the first things that you can check out is what are the core concepts in QIIME2. So there are a lot of things in details that they have mentioned. What do you mean by QIIME2 artifacts? What are the visualizations that are produced, and so on. There is a lot of concepts in QIIME2 as well. Today, I will try to simplify at least the very basic core commands in QIIME2, and so that you all can have a preliminary analysis of your sample someday, right? So let's begin with that. Now, to run QIIME2, you need to have obviously a Linux terminal. You will have to create a mini Conda environment. So these are some of the installation requirements that they're very clearly mentioned. I'm not going through the details of that. So as you can see here, towards the left, there is a QIIME2 environment that has already been activated, right? And I'm using a Jupyter notebook so as to show you all the commands that we can run. Right. So I'll just open my Jupyter notebook. So all that you have to do is go to the required directory, wherein you will be having your input FASTQ files, your metadata file. I'll come to the concepts of that as well. So all of that, whichever directory you will be having those files, you have to go there, activate your environment, which is QIIME that I've named here, and just open a particular corresponding Jupyter notebook. Now, this Jupyter notebook will have all the dependencies already ready for you, right? All the packages, all the dependencies required by QIIME to run will be enabled by this particular, by this environment, and you will be utilizing them through the Jupyter notebook. I'm not going through the details of the installation procedures and all of that because of time constraints. So this is one of the links through which I can access the Jupyter notebook. Right. So here you can see in this particular directory, right, the webinar time to demo. This is the directory that I've created, which I'll be utilizing for this particular run. Now, this directory, whatever are the contents of this directory or this folder, in other words, will be, you can see them here. Right. This is the home page for the Jupyter notebook. Now, this is the notebook for me, wherein I have all the commands ready, and I'll be running them for you. Right. So the Jupyter notebook that I have used here, or I would say whatever is the sample, right, or the input data set that I'm using here is one of the data sets that have been depicted in one of the QIIME 2 tutorials itself. It's referred to as the moving pictures tutorial, right? So you can see what are the details of the moving picture tutorial data set. The number of individuals that they've taken the sample from is 2, right? They have taken it across 4 different body sites, right, from these 2 different individuals, right? And they have taken these human microbiome samples across 5 different timestamps, right? So these are the major details of this particular data set run. And the region of the 16S RNA that they have focused here, or that they've sequenced, is the V4 region. I hope you all remember the variable regions that I showed you all. And here only one particular region, which is namely the V4 region, has been sequenced using Illumina, right? Illumina sequencing platform. So before I begin, the very first step, obviously, in the QIIME 2 pipeline is to import the necessary packages, which is the name of the package here is QIIME 2 in itself. And the second thing, now, whatever commands that you would actually write in a Linux terminal are what is being written here in the format of a Jupyter notebook, right? So whenever you write any Linux command in the Jupyter notebook, it has to start with an exclamation mark as written here, right? So this command is actually a Linux command, but you're writing it through a Jupyter notebook. So mkdir is nothing but for creating a directory. So I am creating a separate directory called QIIME 2 moving pictures tutorial, and cd is nothing but I'm changing my position into that particular directory, right? So let's run this command. First, I'll import the necessary package. To run any particular command in the Jupyter notebook, you can just click on Shift Enter together. So that will run the command. This asterisk symbol here is actually representing that particular command is running. You can also see an hourglass on the top of the tab, and that is also showing that one particular process in the Jupyter notebook is running, right? So let's go to the next command. Now the directories have, this command has been executed. So if you go back here, you can see again that the directory has been created, QIIME 2 moving picture tutorial. And because of the cd command, we are currently within that particular directory, right? Now, let's download the necessary files for the pipeline, right? So there is something called as a metadata file. And this metadata file plays a very crucial role for the QIIME 2 pipeline, right? I guess somebody has raised their hands. I'll be taking questions towards the end, if that's the case. Currently, we'll go through this pipeline and finish it off. Yeah. So, downloading the metadata file, right? Why do we need the metadata file? So you have a particular sample, you have extracted the DNA, you have sequenced it, you have got the FASTQ files, right? Now, we need to have certain informations pertaining to these FASTQ files, right? Like, for instance, which this particular sequences that we have obtained, are they in the demultiplexed format or in the multiplex format? I'll come to that later. What, if they are in the multiplex format, what are the barcode regions that we have used? So there's a lot of information. If we are speaking about a disease versus control situation, if we are analyzing a particular relationship between the gut microbiome and a particular disease, say, so what was the status of the patient, right, of the person, of the sample, right here. So all of that information, any information describing your particular sample is referred to as a metadata file, right? It just aids the analysis. So that is what we'll be downloading here. So here I'm using a wget command. This is to download it from the web page, right? So -o stands for what will be the output file name. And this is the link which I am downloading, the HTTPS link, right? So here you can see that the command is running. This should be done in pretty quickly. Yes, and it's already done. Yes. So this tutorial is tailored in such a way that the commands run quickly. Ideally, this might not be the scenario. As your sample size increases, obviously the run time will also tend to increase for these particular commands. Now, next, so this is the metadata file that we have downloaded. Going ahead to download the main sequence data, the FASTQ files, right, that we require for the run. So these are the two major important inputs for the QIIME2 pipeline, right? So the sequence data and the corresponding metadata. So I'll be creating a new directory once again. So this is to download the sequences into it. So here we go and download the sequences and the barcodes. Now, what is a barcode thing, right? So while this is running, I'll probably shift back into my presentation just to quickly show you all that. So if you see in this particular slide, you can see that there is one red color sample barcode, right? So whenever we sequence a particular sample, we need to add a unique identification tag. We know how different products, we buy different products and each of them have a different barcode. Similarly, for a particular sample, this barcode will act as an identification tag, right? Now, why is this necessary? So whenever we are sequencing on an Illumina platform, it's not necessary that all, say I'm dealing with one particular cohort or a group of people, not necessarily all of those samples will be sequenced together, right? They might be sequenced across different runs of the Illumina machine, right? So we need to have these identification marks so that we can group them together towards the end, right? Their corresponding sample runs. So that is why this barcode is very important. You may or may not have this information. I'll come to that now. So
I'm sorry to interrupt you, Maya.
Yeah.
The thing is, can you just please increase the font size? Because it is
Sure. Is it visible now?
Yeah, it's fine. Yes, it's perfect.
Okay.
Okay.
Thank you so much. I'm so sorry if that was it previously visible, Yasha?
Yeah, it was, but the font size was too small at that time. Now it is fine.
Okay.
Now it is fine.
Okay, Yasha. Okay. Yeah. So going back to the concept of barcodes, like I said, it's a unique identification. Now, this depends on what is your sequencing type, right? So your sequences could either be in the demultiplexed format or in the multiplex format. Now, what do I mean by that? Whenever I have all the particular samples and I already know which FASTQ file comes from which particular sample, that means that the sequences are already demultiplexed. That is, I know that this sequence belongs to this particular person, right? So that sort of an information or that sort of a sequence format is referred to as demultiplex sequences. When I say multiplex sequences, I do not have this particular information. And we need to demultiplex them through the QIIME2 pipeline in itself. That is aided by this pipeline, right? So that is one of the steps here. Now, since we are utilizing the barcodes here, that means that our sequences are in the multiplexed format. And we need to assign the sequences back to each particular sample, right? So we have already downloaded the barcodes. Now let's move on and download the sequencing data, which is the FASTQ files, basically. So this is a general format, fastq.gz, right? This is the format in which we generally get the sequences from the Illumina run as well. Yes, and that's done, right? So all our sequences, so as we can, I'll just go back and show you here, this is the EMP single end sequences directory, wherein we have the barcode file and we have all the sequences that are present. Here you can see that there is only one single FASTQ file, very clearly indicating that your sequences are in the multiplex format. If they would have been in the demultiplex format, you would have multiple FASTQ files. So there is a number limit to that as well, around 2 to 4. So if you have around 2 to 4 FASTQ files, very clearly indicative that your files are still in the multiplex format and you need to demultiplex them. So this is for certain people who are not aware of these particular details or information. They probably do not receive it, right? So this is one particular directory, and this is the major directory that we have already created, and this is a metadata file. Right. Now, beginning with the QIIME2 pipeline. So this is all the preliminary things that we need to do before the QIIME2. Now, the first step in QIIME2 is importing the sequences, right? So any data that is produced as an output from the QIIME2 pipeline is referred to as an artifact. Right? We need to have an artifact in QIIME2 pipeline. There's a very specific term to this particular tool. And why they have used this particular thing is because they do something called as provenance tracking, right? That is, given a particular file, an output file, I know how it was created. What were the steps that were taken to create that particular output file? That is referred to as provenance tracking, right? So to enable that is why they have come up with this concept of artifacts, right? So even when we are utilizing our FASTQ files, we cannot just utilize it in the raw format. We need to create an artifact out of it, right? And to create that is why we use the import command, the very first command of the QIIME tool, right? Now here you can see there are a lot of arguments or parameters that are present in this command. So you can see this is referred to as single end sequences. So I hope you all know that whenever you have an Illumina run, your sequences could either be sequenced in a single end format that you sequence it only in one direction, or it could be in the paired end format. That is, given a sequence, you sequence it from both the directions. In paired end format, you tend to have 2 FASTQ files per sample, right? So here we'll be having only 1 FASTQ file per sample, and that is what has been mentioned through the type, right? You have the input path, right? And this is nothing but the directory wherein we'll be putting our sequences into, and the output path wherein you are creating a QZA file. Now, this is a very important file, which is referred to as the QIIME zip archive. QZA stands for that. So this is also one of the artifacts which are being created through the QIIME2 pipeline, right? So let's import our FASTQ sequences and create an artifact out of it. This type, now there are different formats I would like to mention, add on that. There are different formats in which you can import your sequence in. If it's in paired end format, there is something called as a cassava lane format. There are a lot of formats in which you can import your data in, right? Depends upon your sequencing run, depends upon what information do you have, right? So it's all dependent on you, right? You can decide pertaining to your type of data. Now, the next step that we'll do is the demultiplexing the sequences. Like I already mentioned, we are reassigning the sequences back to their sample so that we know what was their origin, right? So let's run this as well. Now here, you can see that as an information, we need to give the barcode file that we had downloaded, right? It needs the barcode tag so that now it can map it back to each and every sample that is present in our data set, right? So, and this column, this attribute is telling you what column in the in this particular TSV file is containing the barcode information, right? And this is the demux.qza, one of the very important outputs from the QIIME2 pipeline, also an artifact. And this is going to contain all your sequences post demultiplexing. Right. And the last parameter, which is error correction, this is just a log file to tell you the log details of this particular run. Right. So this step may be optional, like I mentioned before, depending on what your sequence type is. Right. So we'll just wait for a few moments so this completes running. Yes. So very clearly, it has mentioned that all the artifacts have been created. Now, one important point that I need to mention here is that the QZA format, right, archive format, this is binary in nature, right? So we cannot, if we need to open the file, we cannot visualize it, right? So for that, there is a command called as summarize. So the summarize command is generally to convert your QZA files to the QZV files. Now, QZV stands for QIIME zipped visualization, right? So this is a format which is human readable, right? So let's convert our QZA file and check out what are the contents of that particular file. So I hope the parameters are pretty self-explanatory. Input data is demux.qza, and now we are producing a visualization of it so that we can view it, right? Now, how do we view these QZV files? So there is one particular website. While this runs, I'll quickly go there. It's called as QIIME2View. Right? So QIIME2View is one of the websites wherein you can visualize all your QZV files, right? So you can just click on this particular web page, and this is just a drag and drop. I hope this is visible. So this is just a drag and drop feature. All you need to do is download that particular file, drag and just drop it here. You can visualize that file. So we'll do that in a few moments. Yes. So the QZB file has already been created. So now if I go here to my home directory, wherein all the contents are pretty much visible, I have to just click on this particular file and click on download, right? Once I download, this particular file will be saved here. Yes. Right. Now I go to QIIME2View. Right. So you can see in my recent downloads, I have the Demux visualization that is already present here. I just drag and drop this. And now you get this particular web page, right? So what is this depicting here? This is just a summary of the demultiplex sequences. So it's telling you how many forward reads were there, right? In our FASTQ files. There are around 1,800 files, right? And this is telling you how many median number of reads are present across your samples. There is a histogram frequency distribution here as well, which is telling you that across different number of samples, how are the different number of sequences distributed. Now, we don't have a reverse read graph here. Why is that? Because this is not a paired end run. This is just a single end run. So we only have the forward reads in this particular part. Now, another important thing that is generated through the Demux visualization is something called as a quality score plot that you can see here. This is a very, very important plot because on the basis of this, we will be identifying certain parameters that we will be giving in the consequent steps, right? Which is the denoising step. Now here you can see that this is the position on your particular read, and towards the y-axis you have something called as a quality score. So the quality score is basically telling you how correctly did Illumina sequence your file, right? Just to put it in very pretty simple terms, right? So it is telling, like if I say what is a threshold quality score, it's around 30. Right? 30. When I say a quality score of 30, it means in around 1,000 bases, only one base is wrong, right? So that's a pretty 1 out of 1,000 error, right? So that's a very good error rate. So 30 is considered to be a very good ideal score. 20 is also okayish, but we generally go with a quality score of 30. Now, anything below 30, right? What do we do is that in that particular read, we don't consider that particular part of the read. We don't take that part, right? We truncate the read there. That's called as truncation. We cut it, literally we cut it in layman terms. We don't consider the information post that position. That position of the read wherein we'll be truncating it is identified by this quality plot, right? So you can even zoom in here. You have to just drag and just hover a bit over there. You can see the different positions, and you can see here, say at position 96, the quality score is around 26, which is below 30, not that good, right? You can see, and post that also, you can see that the quality scores are pretty much declining, right? Below 30. So for this particular run, I have chosen the truncation length to be 120 as mentioned here. Right. So this parameter for the next step is actually identified using the demux.qzv file. Right. So what is this denoising step? the denoising step, which is the next step in the QIIME2 pipeline, is one of the longest steps. So I'll better put it for run. And one of the longest steps in QIIME2. And this is actually going to remove all your duplicate reads that are being generated, any chimeric reads that are being generated. Basically, any low quality reads that are present in your sample are going to be removed through the denoising run, right? This is one of the main features. Second feature is that your forward and your reverse reads, in the case of a paired end run, will be joined together or concatenated together in the data to denoising step, right? So a very important and a very significant step in the QIIME2 pipeline, and also because of that, it's one of the longest steps as well. Now it might run a bit fast because of the lesser number of sample files. So let's wait for that. Now, this is the truncation parameter, like I told you, truncate length, right? So remember that truncate length is a position, right? It's a position on the read, right? Where you want to truncate the read. Trim, on the other hand, trim is nothing but you have a read from the beginning, right? From the 5' end to say. So from the 5' end of the sequence, how many reads do I need to remove? Right? Or how many bases do I need to remove? Not read, sorry. So trim is for that particular parameter. It's the length of the bases that you want to remove, right? Number of bases, basically. And this is also a very important parameter, but this is used because generally in a sequencing run, the quality of the sequencing for the preliminary part is a bit not that good, right? So that is why we utilize the trim parameter, right? So remember, trim is the length, right? And trunk is actually the position of the read wherein you want to truncate your read, right? And there are three important outputs generated here. One of the representative sequences, you have an output table and you have the denoising stats from the data too. There is another optional tool for denoising in QIIME2. It's called dblur, right? You may use any one of them. You can check out the documentation for further details, right? So what does a representative sequence data to contain? It contains the ASVs that are obtained. Like I had already explained the concept of OTUs and ASVs. So this is where it comes into the picture, right? So the ASVs generated are produced through the denoising step, right? So this particular part has been completed. Now we can, there is something called as a denoising stats. So now this denoising stats, again, when you tend to tabulate it, it will just tell you, how many sequences were removed from your file, right? How many of them were retained, how many of them were chimeric in nature. All of that information is present in this particular tabulated form, or I would say the stats, data to stats file, right? So again, as I said, QZA is not human readable. So we convert it into the QZV format using the metadata tabulate command, right? So there are two commands, either summarize or tabulate. These are the ones which we generally use to convert a QZA into a QZV file, right? So let's wait for this to run as well. Yes. So this particular visualization has also been produced. So the next step is that I'm just renaming my files for ease of convenience. I don't want such a long file name. So I've just named it as representative sequences and table.qza. Do remember it's produced from data too, right? Now, the other part that we'll be doing is also summarizing our input table, right? This particular table.qzb into a QZB file, and also the representative sequences.qza into a QZB format, right? So let's run this as well. Again, this is nothing but to convert it into a human readable format, like I mentioned before. So these are the different steps. Like I said, this is, I could call it quality control. You import your sequences, right? You demultiplex your sequences. Following that, you denoise your sequences, right? Remove the erroneous sequences. And towards the end, you do something called as taxonomic assignment to know who are the microbes that are present within our given sample, right? So yes, so it's still running. So I'll probably show you just one more QZB file. So let's go here. So let's see the representative sequences.qzb file. Let's just download this quickly. Right. So, okay, this is the Demux for QZB. Sorry. Right. So here we have the representative sequences.qzb or the ASVs, right? So if you can see here, this is the feature ID. This is an internal feature ID which is being used by QIIME2 internally. We do not have to know about that. Now, this is the sequence of the ASV. Right, this is a representative sequence, right? So this is a very important information, and even we can even blast this particular sequence in the on blast, right? And we come to know what particular organisms are pertaining to this particular sequence. So once again, we are identifying the organisms here itself, right, at this particular sequence, right? So this is one of the a very important file, I could say. That's why I have shown it to you all. And this is also telling you what was the sequence count, what was the minimum length of the ASVs that we found, what was the maximum length. So it's all 120, which is a good metric here, right? It's all constant throughout, right? And different informations pertaining to your representative sequences, right? So let's go back to the command line. Now, the next step is the taxonomic annotation, right? And for the taxonomic annotation, this is one of the last steps, right? Wherein we need a reference database, right? So the one that we're using here is Green Genes, which contains information pertaining to all the 16S rRNA sequences. So this is a reference database against which we'll be mapping our reads, and we'll be understanding what organisms are present in us. So Green Genes was previously used. Actually, the more updated database that I would like to use currently, Silva, has a more better annotated database, I would like to say, right? So for the time being, I'm using Green Genes here. So let's quickly run this. Just a few MBs, and the Silva database is much huger, it's much more huge, than the Green Genes database, right? So for people who have storage constraints, can go up with Green Genes as well. So now we come to one of the most important steps again, assigning taxonomy to the sequences, right? So here the plugin that we use these are called as plugins, which is feature classifier, and the database name that we give here is GG, right? The Green Genes database. So let's run this as well. This is also one of the second longest steps in this pipeline, following denoising. Denoising was the longest one. And following that, we have the taxonomy assignment step, which is a bit longer, right? So let's wait for this. And the output format here will be the taxonomy.qza file. So following this, what we'll do is we'll be converting it into human readable form, just like I've shown here in the next step. Metadata tabulate, and finally we'll be generating a taxa bar plot. Now this bar plot will tell you that across different samples, how are our ASVs distributed, right? How abundant are different microbes across our samples. It's a beautiful visualization, and we'll be seeing it in a few moments. And the time being, I'll explain you the concept of plugins, right? So QIIME utilizes something called as plugins. So this is for the developers, right? So there might be different developers across the globe who are interested in contributing to QIIME2 as a tool. So they come up in the QIIME2 forum. If you check out, different research scientists come up with different plugins, right, to aid different features in metagenomic analysis and such, right, for 16S RNA analysis as well. So feature classifier is one such plugin. These are called as plugins, right? So they are not something which is internally present in QIIME, right? And it also doesn't depend upon what language you're writing the plugin in, right? So it may be R based, like for instance, the data to denoising step, right? So that particular plugin is written in R, right? But QIIME in itself is written in Python, right? So even it's somewhere aiding cross-platform based analysis as well. So this is one of the very cool features of QIIME once again. Yes, so the taxonomy.qza file has been generated. Now we convert this once again into a QZB file, and using that, Now we can generate the taxa bar plots, and we'll be visualizing them. Once again, you can see here how the metadata tabulate command has been utilized to convert the archived format into a visualization format. So the last step of the QIIME pipeline. Yes. So now we generate the taxa bar plots, right? So you can see how we have given the input as table.qza. This is the table which contains your ASV sequences, right? You give the input taxonomy that was generated through the classifier that we used previously, right? And you also give the sample metadata file. Why is this important? So that we know, suppose you have a division within your data, right? So there we can know, like suppose we have a control group and we have a patient group, we can identify what are the differences in the microbial distribution among these two set of samples, right? So that is why this is very important to give the metadata file. Now let's visualize the taxa bar plots. I'll download the taxa bar plots here. Just click on the download button, like I said before. Go to the QIIME view page and upload your taxa barplots.qzb file. Okay, yes, that's done. Right. So this is one of the taxa bar plots that you can see. Probably I might zoom in a bit. And there are different taxonomic levels at which you can visualize it, right? So this is level 1. Now, level 1 implies kingdom, right? I know you all know the taxonomic hierarchy, right? So the level 1 will be the highest in the taxonomical hierarchical order, starting with kingdom. Now, when I go to level 6, this will imply a genus level classification. So you can see here, K stands for kingdom, P stands for phylum, C class, O order, F family, and G stands for genus, right? So you can see how the different genuses are present. Just to zoom in so that you all can see here, you all can see these are the sample IDs, right? And towards the y-axis, you have something called as a relative frequency. This is also a concept in detail. I'm not going into the details of it, but you can see what is the percentage of the ASVs that are present and how abundantly they are present across different samples, right? So here you have the genus level classification that has been shown here. If I go to level 7, I'll get right up to the species level, right? A bit limited because of the 16S rRNA sequencing. If you would have gone through the shotgun metagenomics approach, you would have gotten a much better resolution, right? So there are different tools for their analysis as such. So you can see here, and these are the legends for each. They have all been color coded. You can see here, and very important. And also you can download this particular information as a CSV file, wherein your rows will be your different sample IDs and your columns will be these different taxonomical annotations as shown here in the legends, right? And you can see how abundantly these taxonomic groups are present, right? So I guess with that, we have come to an end to the QIIME pipeline, right? So these are the very basic commands that I've covered for a very preliminary analysis, right? And I encourage you all to, go ahead, read the documentation further. And there are a lot of things that you all can do with QIIME. There's a lot of statistical metrics also that can be computed through QIIME, which I've not covered today because I'm out of time already, right? So yes, that's it from my side. Yasha, probably over to you.
Yeah, thank you. Thank you so much. Okay.
Yeah.
So, thank you very much, as your presentation was instrumental in shedding lights on the importance of microbiome and clarifying concepts within metagenomics. It was truly fascinating for me, and I'm very sure for the viewers to explore the shotgun metagenomic techniques, unravel the intricacies of 16S rRNA gene structure, and the comprehensive understanding of QIIME pipeline along with its hands-on guide. So thank you so much. Your knowledge and insights have been greatly valued by us. So now we understand that our audience might have some remaining questions that we'll address now. I warmly invite our audience to post their questions in the comment section pertaining to the topic discussed. So we'll take the questions now.
Okay. Yes, I'll be sharing the presentation definitely. How to download the QIIME2 files. I guess you're asking about the input files. So this depends upon your sequencing run, right? You need to have a sample for that. You need to sequence them using the Illumina machine, and you'll get a set of FASTQ files. And on the same run, you'll also get a corresponding metadata file, right? So these are the two main inputs for the QIIME2 pipeline. Sudhanva, if I answered your question. Dr. S. Karthik, you have asked what are the main differences between QIIME1 and QIIME2. QIIME2 is a much more advanced version of QIIME1. First of all, there were a lot of commands in QIIME1 which were not, which didn't have that much of utility, if I would be precise. A lot of improvements in QIIME2, and I encourage you to definitely use the latest version. Yes. Okay, regarding sharing the code, yes, I'll definitely let you all know, but please don't worry regarding the code. Detailed tutorials that are present on the QIIME2 webpage itself. So if you all are stuck any point of time, you all can definitely refer the documentation for that. Yes.
There's some question on, can we implement this pipeline using Kraken database?
Yes, so Kraken is used for shotgun metagenomics. It's a tool used for shotgun metagenomics, not for 16S rRNA-based approaches, right? QIIME2 is specifically used for 16S rRNA. Yes, Sangeeta has asked, what is a primer?
Yeah, in that slide that you had talked about the barcode primers along with the barcoding.
Primer.
Yeah.
Yes. So a primer is a sequence. So there is, I don't know if you're familiar with the PCR, the concept of polymerase chain reaction. And, whenever you have a particular sequence, when you deal with these, say, microbes, right? Now, the quantities that you're dealing them with will be really very small. Obviously, they're very microscopic. So to, even the quantities that you're dealing with will be very small. You need to amplify the amount that they're present in. That's why we use a PCR. And a primer is just one of the sequences. It's a short sequence which we use that will be complementary to your target
template
sequence.
DNA. Yeah.
Yes, to your template DNA, and you will be utilizing it to amplify your sequences. I hope I clarified your concept. If an individual is
occurring again and again from HP, is how the sequences from PacBio or Illumina platform are imported to QIIME2. Also using multi-QC for quality control is a better approach or not?
Yes, you should definitely follow it with, see, there are a lot of quality control checks that internally happens within QIIME2 itself. But yes, it's always better if you go with tools like FastQC and multi-QC, wherein you'll be having a preliminary check of your sequences. And initially itself, you can cut down on the number of RNA sequences that might be present. And how are the sequences imported? Definitely, these runs, particularly, they have a server wherein you upload all your files, right, from the PacBio and Illumina. You download it onto your system, and then you need to import it using the commands that I just mentioned in the QIIME2 pipeline. I hope, HP, I've answered your question.
Yeah. Another question that they're having is, since we only talked about microbiome, S. Karthik is asking that, is there any specific database available for microbiome, that is the fungal community?
Yes. So in the case of microbiome, we use something called as ITS sequences, right? So there definitely has to be a database for microbiome sequences, although that I am not familiar of, but you could definitely check it out. Out of my knowledge currently. I'll definitely get back to you if possible.
All right. Next, from Manoharan, we have is, how do we decide the read length, the total length, and the data quality for the soil sample? I think it's one of the most important questions.
Yes. So whenever you're going through an Illumina platform, right, definitely the read lengths are, around 150 to 300 base pairs, which is a very good read length, right? So anything around 150 to 300 pretty good quality, right? Total reads will depend upon your sample. It depends upon your sequencing depth. There are a lot of parameters that you need to check in the Illumina run itself, right? And data quality, that will depend upon your data, your sample preparation, right? How you deal with your sample. Also, a small amount of errors are but obvious because the steps in itself are such that you may introduce certain errors into your run. But that's why we have so many quality control steps in itself, right? So Sharanya, yeah, I hope I answered your question.
One more thing, Maya, I think it is also important because it's one of the most famous questions most of us might be having in their minds. So Devrit Gupta is asking that, Devrit Gupta is asking that, then if you're not from bioinformatics background, so how can they do this? How can they learn this? How difficult it would be, or what approach they should follow?
Yes, so that's an interesting question. Certain basic biology-based concepts are important for this. I would not deny that fact. Say, you need to know what is a 16S RNA molecule, what is a ribosome, and all of that. You need to know what is an Illumina sequencing platform, what is a FASTQ file. So these are certain very basic concepts. But even if you're not from the bioinformatics branch, I feel if you learn these very small core concepts, I'm pretty sure you can deal with this. It may seem complex in the beginning, but it's not that complex, Devrit. So, yeah.
Yeah, thank you, Maya, for that. And people are also asking about the applications, as in, is it useful for, in what ways is it useful for bioanalytical sciences and other fields of biotechnology?
Yes.
On that.
Yes, very, very good, important question. So I will probably brief regarding my current project, which is going on. So what we do in the healthcare domain, right? So there's a lot of diseases for which there has not been I would say, a very solid cure yet, right? And there is a lot of correlation of your gut microbiome with different parts of your body. Okay, one important such I would say relation is between your gut and your brain. Okay, a very, very important. There is something called as a gut brain axis, right, wherein the gut microbiomes can have a direct influence on your brain, right? So many particular disorders, say for instance, you're talking about Alzheimer's, you're talking about Parkinson's, right? These are certain disorders that scientists have been breaking their heads to find a cure for, and now people are moving towards gut microbiome based approaches for finding a cure for such diseases. Some other diseases include stroke and diabetes mellitus. There are a lot of such diseases for which they seem to find an implication by the gut microbiome, right? So this is one of the main applications that I would like to tell you all. Second of all, there is also a concept called as personalized medicine or precision medicine, right? Wherein customized, so every individual will have a different gut microbial distribution, right? Or any microbiome distribution for that matter. So very much customized medicine for that particular person, right? So the world is moving towards that, wherein specific to a person's microbial profile, you have certain medicines that are tailored to them, right? So this is also one particular field which is coming up. And also now the trend is for multi-omic based approaches. So combining not only metagenomics, you can combine it with a lot of other things. There is metabolomics, metatranscriptomics. The field is just endless, right? So yeah, some of the very few applications, if I could brief upon. Yeah.
Thanks, Maya. I would request participants that if any more questions they have in their mind and they think it is important, they can unmute themselves and ask. If not, then we'll move ahead.
Hello.
Hello.
Hi.
This is Raj Oja from this side. First of all, I would like to thank Maya Madam for such a good session on QIIME. And I have a question, Madam just quoted once, using database between green genes and saliva. Right. I have a question there. I think if I'm not very sure about this, but Silva resolves up to genus level of rank, and
Yes.
green genes has a species level up to resolution into the comparison between bacterial species.
Yes.
First question is about that. Like, if we are getting more updated database such as Silva, then we are compromising the resolution of the bacterial species. While
Yeah.
if we are using green genes, then we are compromising the updates, as you quoted. Also, I would like to ask, can we use RDP here as a reference database? Thank you. That's
Yes.
from my side.
Yes. So, answering to your first question, depends on your project and depends on what is your focus, right? So, if you are okay with compromising on the taxonomic classification, then, like you said, you could probably go ahead with Silva, right? Why I would prefer Silva is because I said it's a better annotated database. That means much more sequences are included in that particular database. So certain organisms that might be missed by green genes might be annotated using Silva, right? That is where the advantage comes in. And for certain sample runs, you might have to specifically focus on the organism as such, right? Wherein you need the specifically the species level annotation. Under that scenario, probably you could go with green genes. I'm not saying green genes is bad, but yes, it's not up to date. Is what is the information that I have read as per my knowledge. And question 2, I think you asked regarding the RDP database, and I think yes, you can also use the RDP database. But I don't think I've heard of situations wherein they've used it within the QIIME2 pipeline. Generally, they go with green genes or Silva. So yeah, I hope I've answered your question.
Yes, thank you.
Yeah.
Maya, there was one question from Manu. She's asking about while you were running the QIIME pipeline, the hands-on part, where she's saying if we have demultiplex the data, will the denoising step still help to download the demux.qzv file?
Is it the demux.qzv file that you mentioned?
Yeah, yes.
Yes. So no, in such situations, we do not require, if I don't know if I should go back to my notebook once. Okay, I'll just say it orally. So, there is a demux summarized.
I can request, if it would be easy, I would request Mahesh if you could help her display her own presentation.
Yes. Okay. Share my screen. Is this visible?
Not, yeah, now it is. Now it is.
Yes, right. So here, if you go through this, I don't get this. Yeah.
Just zoom in a bit, Maya. Yeah.
Yes. Is this fine now?
Now it's perfect.
Yes. So if you go through the QIIME2 documentation as well, you will see that this demultiplexing the sequences step, like I mentioned previously, it's not a necessary step, right? Depends on what type of sequences like you have. So if you already have demultiplex sequences, you can you have a step after the importing step itself. You can directly go to the denoising step. Right. So there is an advantage if you have the multiplex sequences already. Right. So yes, that's an advantage. You can skip this particular step. I'll also show you the documentation here. There are a lot of Jupyter book tutorials. There are normal tutorials as well, which you can go through, right? And they're very clearly explained all the concepts. It seems to be a bit vast, and it is, but it's not that hard, right? You just spend some time in reading them. So yeah.
Okay. Thank you so much, Maya. Thank you for addressing all the questions patiently. So before wrapping up folks, there is an amazing news for all of you. Soon we are going to conduct a free virtual internship on microbial genomics for beginners. The details will be shared soon on our social media sites and on the link shown on the site. So please stay tuned for the details of the internship. So as we move towards the end of our webinar, I want to express our deep appreciation to our wonderful guest, Ms. Maya Jayachandran, for generously contributing her time to this event. Her insightful journey through the realm of 16S metagenomics using QIIME2 pipeline must have been a source of inspiration for many students, young scientists, and researchers. We have faith that our audience has acquired a significant amount of knowledge. Dear audience, you'll soon receive a feedback link from our Instagram team. Please make sure to provide your current email and name when completing the feedback form. Additionally, we kindly ask you share your certificates on your social media platforms, tag us, and help us spread the word about our future events to potential participants. Before we are signing off, I'd like to extend my gratitude once again to each and every one of you and offer my best wishes for a fantastic week ahead. Hope to see you in our next webinar soon. Thank you. Bye-bye.